Hello, everybody. Today we're going to talk about desiccation tolerance. According to Wikipedia, desiccation tolerance refers to the ability of an organism to withstand or endure extreme dryness or drought-like conditions. Of course, we're going to talk about plants. So this is an amazing plant called Praetiostigma plantaeogenium. On the left, we can see this is the fully hydrated state, where we can see all the leaves are spread out and we can see a flower growing over there. But after we remove water from it, we can see the leaf is starting curling up and it's shrinking, which forming this kind of like weird shape. But after we add water back into this plant, it forms the leaves back again and also the flowers going back. This kind of plants, we call them resurrection plants. So what do I mean by resurrection plants? Actually, resurrection plant is just plants that can go back to their original states after desiccation. But non-resurrection plants, they're just gonna go death after desiccation. But why we're actually going to study plant desiccation tolerance? Because we can identify mechanisms that can be translated to withstand water deficiency in agriculturally relevant plants. And we can discover the metabolic or molecules with beneficial properties and understand the metabolic reaction in non-aqueous cellular compartments. So this is a normal cell where we have the nucleus, the mitochondria, and some chloroplasts. But the vast majority of this plant is still going to be the central vacuole, which is mostly component of water. During desiccation, we know the water is going to be lost. So how is the plant going to look like? And how is it even able to go back to its original states?
Now, let's dive into this fascinating aspect of desiccation tolerance in the journey from survival to death under drying conditions. This is the general vegetative drying curve that intuitively shows you a continuous change of survival to death in conditions in plant cells. Plants are classified as exhibiting vegetative desiccation tolerance if they can survive drying to a leaf water potential of approximately negative 100 megapascal in MPa. The blue curve is the drying curve for a plant cell undergoing desiccation, and the dots here are approximate water potentials at which responses occur. As cells lose water, they undergo mechanical and structural stresses associated with decreases in volume and increases in cytoplasmic compaction and viscosity. Cells lose turgor as they dehydrate to approximately negative 2 MPa. Vegetative cells decrease in volume by 60 to 80% as they reach negative 4.5 to negative 6 MPa, which imposes the physical strain on the cell wall and plasma membrane as well as areas of attachment between them. As water potentials are reduced to about negative 5 MPa in vegetative tissues, vesicles must vesiculate to avoid rupture. Here, mitochondria and chloroplasts lose their shape and internal organization when water potentials reach negative 5 MPa. So importantly, the key problem of desiccation is that as cells shrink, their cytoskeleton is disrupted and cell contents are brought into juxtaposition, resulting in interactions. As water potentials continuously decrease, membrane fusion and loss of cellular compartmentalization can increase, resulting in cell death. Okay, so first up, let's chat about how these plants get ready on the metabolic level. As cells lose water, the primary threat is to the integrity of both their organelles and plasma membrane. Desiccation-tolerant cells protect them in several ways: through modification to membrane composition, through employment of efficient antioxidation mechanisms, and by non-reducing sugar-mediated stabilization. The plant cells stock up on certain sugars, amino acid, and antioxidants, even when they are not dry yet. Think of it like a plant version of an army, all geared up and ready to face the dehydration battle. This doesn't just slow down the loss of water, but also acts as a shield against these nasty things called reactant oxygen species. ROIs are harmful molecules that increase when plants dry out. These plants have special defenses, like antioxidants, to protect against ROS damage and fix their cells. Now, as you can see here, how do these sugar and amino acids actually help? They work as osmoleates, which is the fancy way of saying they keep the cells plump and the space in the membrane just right. So, Everything holds together well. Molecular crowding during water loss increases the probability of the cytoplasmic cells interacting with the protein surface. This is super important because keeping the plasma and all those tiny organelle membranes intact is key to stopping the cells from totally collapsing when they dry out. As you can see here, in the intermediate water range, we can see the presence of preferentially excluded cellulose intolerant cells keep the membrane and keep the membrane surface preferentially hydrated and prevents the cell membrane fusion. The absence of these cellulose in the sensitive cells might result in membrane fusion as in model membrane systems. Let's go into the protection during dehydration. There's another cool part. There are two strategies. The first one, preservation of cell turgor and membrane integrity. The second one, transition of cytoplasm to a glassy state, preventing cell damage. As the plant dries out more, the stuff inside the cells, the cytoplasm goes from being liquidy to this glass-like state. This is called vitrification, and it's like adding an extra layer of armor to the cells, keeping them from getting squished or damaged further. As the cell's potential drops toward negative 100 MPa, sugars contribute to the transition of the cytoplasm from a gel-like matrix to a gaseous state. Vitrification in plant cells during desiccation happens as follows. When water loss, as the plant loses water during desiccation, the concentration of cellulose in the cytoplasm increases. This increase in cellulose concentration, like sugars and proteins, leads to a higher viscosity in the cytoplasm. As the viscosity increases, the cytoplasm transitions from a fluid liquid-like state to a more solid glass-like state. The glassy state of the cytoplasm provides mechanical stability to the cells. It prevents damage to cellular structures by limiting the physical and chemical changes that can occur during the severe dehydration process. And when it's time to rehydrate, it's not just the simple soaking up water deal. The cells get busy fixing and rebuilding any bits that got damaged. The antioxidants go into overdrive to handle any oxidative damage from those ROS deals, making sure the cells don't just make it through but actually come back stronger after the whole drying out episode.
Our journey into the heart of plant resilience through the study of desiccation tolerance reveals the critical roles of ABA and ROS are key players in the plant survival mechanisms. ABA's roles in enhanced drought resistance through the water conservation and cellular protection mechanisms, alongside ROS dual functions in stress signaling and defense, underscore the difficult balance plant maintain to survive desiccation. Research in the field have made significant strides, identifying the key players such as LEA proteins, antioxidants, and various stress response genes that keep plant survival under the extreme dehydrations. These discoveries are not just academic, they hold a promise for practical applications, particularly in enhancing the agriculturally important crops. By manipulating this gene pathway, scientists armed to develop crops that can withstand the growing of the climate change, ensuring the food security for the future generations. Looking ahead, the futures of desiccation tolerance research is bright illuminated with the probabilities. The next frontiers involve not just furthering our understanding of the mechanism withstand the desiccation tolerance, but also applying the knowledge in innovative ways. Advance in genetic engineering and biotechnology offer the exciting opportunities to transfer desiccation tolerance traits to crops, potentially revolutionizing the ways we approach agriculture in arid or semi-arid regions. As we stand on the cusp of this advancement, it is clear that the study of the desiccation tolerance is more than scientific progress. It is a crucial step toward sustainable futures. By harnessing the powers of nature's most resilient plants, we can pave the way for a new era of crop production that both resilient and resource efficient.